Good morning, everyone. Thank you so much for having me here. I'm delighted. This is a room clearly with a lot of interested people, a lot of energy. This is terrific. I am thrilled to be with you and pleased to have been given the opportunity to pass on to you what is most urgent for our times. And I hope that this session will serve as an opportunity to begin thinking differently. And I'll show you how you are going to be so important for what is to be the changed in the phase of what we call Parkinson's. So thank you, Irene, for such kind introduction. Thank you for organizing all of this. And my visit yesterday at the University of Florida was wonderful. So I'm delighted to have had a time to meet with terrific colleagues. You guys have a wonderful center here in Gainesville. It's one of the world's best. And I'm just thrilled to be here. It's funny to be in this slide because this slide is showing tombstones. And what I wanted to tell you is we have to change. There is no Parkinson's disease, but people with Parkinson's. And what we're studying heavily is Parkinson's disease. We are experts on Parkinson's disease, which means that we are not experts on what disease somebody with Parkinson's has. And it is not about Parkinson's disease, but about who has Parkinson's disease. That's the main message for today's session. And I'll show you how important that is. First, by beginning with the idea that we have been trying to change this disease and are going nowhere. Because if we think we know something about Parkinson's disease, we think we know something about people with Parkinson's disease. And then what happens to be success in a laboratory animal, we figure it has to work in people because that animal must tell us all we need to know about people with Parkinson's disease. So we do these clinical trials and they go nowhere. And they are fantastic drugs. Creatine, coenzyme Q10, glutathione. There's still clinics of glutathione in this state, by the way. We don't know who those drugs really work for. But when we give them to Parkinson's disease, none otherwise specified, they do not work. We have a pipeline. Up on the tube and the top two, those have already become tombstones. We just haven't seen the reports. So Parkinson's is, we say to ourselves, heterogeneous. So when you are in the clinic, your doctor would have said to you, based on all I know about you at the bedside, you have Parkinson's disease. Trust me. I'm an expert. And not only that, I know the range of Parkinson's, and I know that Parkinson's could start with a bad sort of presentation with very rapid accrual of milestones, but we also know that it can really go all the way to this other end of the spectrum and be malignant. But we know that this is Parkinson's in general, and I've lost control. That the issue here, yeah, there he is. Thank you. So all of these patients, we think, are Parkinson's because, and you'll see in a moment why we think so, but largely because we are the experts. So we hasten to say, sorry, we don't have a test to prove it to you, but trust me, I'm an expert. It's great, great medicine for the 19th century. All right, so this is part of the problem. the way we understand Parkinson's is forensic. Essentially means we have a fire going in a metaphoric way, but we cannot do anything about the fire because we can only sample the fire from the outside. We sometimes do spinal taps, we check the blood, see if we find something, we do skin biopsies. There are some biopsies done on the submandibular gland, trying to find evidence outside. But really what we're trying to do is saying, trust us, you have Parkinson's, because if we wait till you die and examine your brain under a microscope, we would have proof. We have proof that that's Parkinson's, right? That's forensic medicine. That's what it is. So here's where we are. We're scavenging on the ashes of a fire that passed. And when we find stuff that we think is what we were expecting based on what we know, we say, number one conclusion, I was right. You have Parkinson's. Have the evidence now. We got it. But you have to say this to the family members because, of course, unfortunately, right. But here is where you have to help us change, because this is what we say too. We have found the arsonist. That stuff that we attribute the association with Parkinson's caused Parkinson's. Gigantic leap of faith. It is belief without evidence. And I'll show you evidence that there's a lot of evidence against it, but when you fight against belief, the evidence does not win. And it is going to be up to you, because we neurologists are the most protective of medical species. It really is. We think we know the truth. Other fields of medicine have used technologies to probe living biology, to question everything they've ever believed. We are true experts. We're the most intelligent of all medical doctors. And so we're using technologies to probe biology to validate that which we know to be the truth. And the truth is that what we find at the ashes of the fire caused the fire. Because of it, the universe of what we call Parkinson's is expanding, is growing. This is considered the largest growing disease, because just about anything that looks on autopsy to be Lewy body, we then think, oh, that stuff, that must have been Parkinson's disease. This is an actual figure of a paper in 2014 that used the best analytic technologies to show us where each of the things that cause Parkinson's map out in a cell. And if you look through it, you can have Parkinson's disease with a damage in just about any part of the cell. So of course, if I'm a company and I have a product that does something related to protein degradation systems, I say, I think I have a treatment for Parkinson's disease. Part of the theme of this presentation is to convince you that there is no Parkinson's disease, that there is people with Parkinson's disease, and they are different. And what we're creating are averages. This is 2014 state-of-the-art knowledge about Parkinson's disease. It may not apply to anyone. Averages are averages. They're not going to ever make you exactly the one that would have qualified for this. So this is essentially what we're saying, that Parkinson's is a puzzle and that we're just so close to completing that puzzle, right? That's essentially what we're saying. There might be other pieces around, but we probably are pretty. Actually, there's an interview by a very famous neurologist on the internet, and he starts with the analogy of the puzzle, and he says, back in the day, we have so very few pieces. Now, you know when you build a puzzle that you start with the corners. We got the corners figured out. That's what he said. This was just a year ago. This is how we think. This is how we think. We think this is the puzzle, and we think that these are kind of there. We may have a few other pieces, right? We think that they kind of fit together, but in reality, on close inspection, you know that we're trying to force the pieces into one another. That's really what we're trying to do. Now, if you look at this really from a completely different perspective, you say, is this a puzzle, right? Or could it be that what we're dealing with is really more than one puzzle? We're really dealing with many puzzles. Isn't it possible that each of us is a bit of a puzzle that wouldn't really quite jibe with others? So if you have this many, it's probably likely that in a future, there wouldn't be such thing as puzzle, but in fact, this range of puzzles implying a range of Parkinson's types, such that then you would imagine that each of them would be amenable to therapies that would be specific for this. So what would a neurologist in the future say when talking to a patient? And this is my hope. Based on all I know about you today, you have Parkinson's disease. No idea what it means to you. We will have to do this test, this other thing, we've got to do that other test, and then I'll know what disease you have, and then we know how to treat you. Meanwhile, we can start with a little bit of levodopa to make you feel better. That is precision medicine that every field of medicine other than neurology and psychiatry have adopted. But because we know what Parkinson's is, why do we question it? We know. We have a great puzzle already built. We have the corners figured out. All right, so if we separate out this puzzle into these pieces, as you see here, it is possible that these are the beginning of individual abnormalities, but we want to see them coming together. And they come together into a pathology that we call Lewy pathology or synucleinopathy, because these Lewy bodies that define what Parkinson's is made up of synuclein. So that's why it's called a synucleinopathy. And another convergent element is dopamine loss. It is very eloquent that the dopamine deficiency makes people slow and the dopamine replacement brings their speed back. So that's good. When levodopa, which we all make, was discovered and made into a therapy, it was a miracle. And we thought the disease had been conquered. But we were doing replacement strategy. And levodopa is to Parkinson's what insulin is to diabetes. But there are specific subtypes of diabetes, you treat them differently. But this is a replacement strategy, a wonderful replacement strategy, but a replacement strategy of what is convergent. So there are things that are common to people, can help us on symptomatic treatments. And all we have available, as you know, are symptomatic treatments, because disease-modifying treatments have never been developed, not for lack of trying, as I showed you in the tombstones, but because we think we know what the disease is in people with the disease. So when you go to this model, you're essentially saying, If I find somebody with anything related to mitochondrial, that's part of the Parkinson spectrum, and therefore I know I can use that as a way to treat everybody with Parkinson's because it's telling me something meaningful about everyone with Parkinson's. And of course, we think though that it's going to reflect something meaningful about what we know about the disease in everyone. And so here's the model. We have this, turns out, inconveniently, that having just alpha-synuclein Lewy bodies in Parkinson's is the exception. The rule is you have a whole bunch of other things. And in fact, because our model predicted that the only way to have Parkinson's is with Lewy bodies, we have a thriving line of research on copathology. Meaning, why is it that these people have these other pathologies if we know they shouldn't have because they have Parkinson's? I said they had Parkinson's. So there are many artificial lines of research on which millions of dollars are invested to try to figure out why this is. It should just be this. So we have someone that let's say one of you is especially in the way that we can figure out what is it that actually happened at a molecular level, at a biological level. And the way we would report that in the literature, because let's say that it's novel, we would say we've uncovered a new target for therapeutic development in Parkinson's disease. It sounds so good. Journals will love it and they will publish that paper. We've essentially found another piece in the puzzle of Parkinson's, and this might in fact be the most important piece in the puzzle of Parkinson's. So because of that, then we're going to develop the therapy, and in fact we may already have a therapy that does exactly what we think should do for that specific pathway. And the way we do it is, as probably you now anticipate, is that we give it to everyone. No matter what piece of the puzzle you're fitting into the model, you're going to get that one, right? And we know what happens to that. So back in the 1700s, there was this traveling scientist. At the time, I think the term scientist might have been a little bit too much of a... But he had a mechanical duck. This was Jacques de Vaucanson. He had a mechanical duck. And he would bring it to shows around the country to demonstrate something that he as a scientist had discovered, which was that the digestive mechanisms can be essentially demonstrated to occur if you have the right structure, you have the right levers and bolts and nuts and all these wheels. If you have all of that moving, you can demonstrate that you feed that particular mechanical duct and you would get the droppings right here. He then showed this duct at different demonstrations. The duct would be sitting here. Let's say that I'm Jack the Dawkinson and I just pull from my pocket, just get my seeds, give it to the duck. And then, meanwhile, let the duck do its thing. And here's sideshows everywhere. Over there, there's dancers. Over here, there are other people just doing their show. Just a beautiful event with great food, all sorts of beverages. In about an hour, you'll get your attention back to me. Hey, let's figure out what's going on. Lo and behold, he had collected droppings, demonstrating the power of science to show that we can reduce the complexity of digestion into a model. Nobody, of course, knew that he had brought his own little bits of droppings in the other pocket because everybody's distracted with the shows on the side. That's Parkinson's. We have a model of Parkinson's. There is no Parkinson's, but people with Parkinson's. Not too long ago, there was something very similar. We were thinking Parkinson's as an elephant. And we're thinking, gosh, if we just need to put it all together in a better way. Some people are thinking it's a problem with the smell. Some people are thinking it's a problem with the autonomic system. Some people are thinking it's a motor problem. Something, right? Maybe it's all the whole thing together. And a lot of what we've been doing is trying to create this compelling picture of what Parkinson's is, because we feel more comfortable when we make sense of the world's chaos, right? Yeah, you look a little bit different, but trust me, you have Parkinson's, right? Trust me, you have Parkinson's. So in reality, you look at this and everything that could happen to a duck, and let's keep on the analogy of duck. In reality, a duck is made of different things, right? Things that are related to genetic abnormalities, things that are related to clinical abnormalities, to a whole bunch of biological factors, proteins, etc. So you can imagine that there is a proverbial pond that creates the duck that you think is the duck. But is it the duck, or could you say possibly that there are several ponds and several types of ducks, and that you could potentially recognize the duckness of them all without saying this is a duck, right? You probably begin, well, this one is a little, right? So you can begin to imagine that Parkinson's is not this one multi-headed beast, but in many ways a collection of distinct animals, each of which depends on different factors that make us who we are. We are all at the end very unique. So is our Parkinson's. Now, but that's not how we're doing science in Parkinson's. Remember, we doctors own the truth. So the truth is that we can say who has Parkinson's and who doesn't, right? Not only that, we're so sophisticated that we can say who has Parkinson's with tremor and without tremor. And we have a very good name for the tremorless Parkinson's. We call it postural instability gait disorder, PIGD. So tremor and PIGD. These are the two types. These are the truth in a literal sense, because when we developing biomarkers, what we do, we start with the truth, which is this. Then we look for the technologies that probe into biology to measure as much stuff as we can. And then we ask a statistical question of all the stuff we're measuring. Which ones correlate with this group, that doesn't with this group, and in the process, we call them, oh, biomarker. That's a biomarker. So we're asking the phenotype, the way we call things, to be the gold standard against which everything else is validated. So how are we doing with this? Well, let me show you how we're doing with this. These are two large multi-million-dollar biomarker development cohorts. And I'm not going to go through this data in detail, but I'm going to tell you that not only could you not use any of this data at all, you cannot imagine a future in which you say, oh, let me just check your phosphorylated tau to understand the kind of Parkinson's you have. It won't happen. Because look at that spread. It turns out that what truths we're validating here, you cannot validate them in the other study. So there are inconsistencies between cohorts. So if we're finding biological truths here, because statistically, these three compared to controls are lower, so we call them biomarkers of Parkinson's in that study. But look at this. Statistically, only this one correlates in this study, not the other ones. The other ones fell off the statistical significance bandwagon. So there are also inconsistencies within cohorts. So if you do this analysis a couple years later, you get different answers. Now, if anybody dies over the age of 85 or 90, and let's say has anything close to what we call Parkinson's or Alzheimer's, we no longer say, as we would earlier in life, oh, had these people lived long enough, they would have developed, right? They are already the centenarians. So there are studies on centenarians that are really meaningful because people live as long as they did, longer than a normal lifespan, with the stuff that we use to classify diseases. So this literature, the literature on the oldest old among us, is filled with the word paradoxical. Look at this to begin with. Half of us meet criteria for Alzheimer's pathology. They should have it because they have everything in their brains. But they don't have Alzheimer's. That's so paradoxical. The numbers are a little lower with Parkinson's, still a gigantic number because we're talking about people that by then should have whatever we had predicted that they would have. How could it be that you can have normal cognition and have more Alzheimer's pathology than if you have bad cognition? That's paradoxical. And if you have a protective form of apolipoprotein, that is associated with more of the bad stuff because it must be bad. It's so bad that in papers we call these toxic proteins. We attribute the word toxicity, and there isn't toxicity now proven by biophysical experiments of any of the proteins we use to classify as pathogenic. So why do we keep failing? Well, number one is because we have great therapies, wonderful treatments, but we don't know who we're targeting with them. We just are not matching what could work to who will be best targeted for that to work. Because the biomarkers we're developing are not biomarkers to understand people with Parkinson's, are biomarkers to understand Parkinson's. We need divergence biomarkers. We need the kind of biomarkers that say, if they're present, that will tell me something about you and about very few others. So biomarkers of divergence will be present in some, absent in most. But that's not that sexy, right? Because we don't understand Parkinson's disease. We want to uncover new therapeutic targets for Parkinson's disease. So obviously, how do we improve? We need to match the what to the who. We really did. When companies are coming to us and saying, we have this fantastic product in the pipeline, it's killing Parkinson's, not the mice. Rescuing the mice, killing Parkinson's. Doctor, please help me bring this to trials so that we can get it to the market. Plenty of that. That's what happens all the time. The question should be, tell me again how this works and therefore who might that be for? We're not asking that question. The who is missing. Cancer recognized the importance of the who for a long time. They essentially underwent a major crisis in the late 70s and 80s when they were saying, look, we're going to cure cancer. And then we're going to cure cancer. And they realized, all right, they had a major failure. They could have said, gosh, no, I think it's the trials are not really good. We need to do more trials. Which is, by the way, we say that, right? Because we own the truth. When we have negative trials, we question the trials. We don't question our truths. Our truths are perfect. So they have long realized after their own difficulties, after their crisis, they learned from the crisis that there wasn't cancer, but people with cancer. And that people with cancer were different, one from the other. It was a revolutionary concept in 1981. And that's what that. So we need to change from cancer to Alzheimer's to Parkinson's to all brain diseases of aging. It's the same thing. Biology did not skip the brains only because we think we are so smart. At the end, we need to understand aging. That's our moonshot. Aging. Why are we aging in funny ways? Some of them funny that we call them Parkinson's. Some of them funny, we call them Alzheimer's. Some of them funny, we say, oh, it's a mixture between Parkinson's and Alzheimer's, right? But instead of going from what we think as the truth to what is, in fact, the biology, we revert it. We revert the order of development. So we first look at the biology, then determine what are the outliers, particularly those for whom there may already be a therapy. And then we say, look, we're finding molecular subtypes, biomarker-driven subtypes, biotypes, right? Because you want to understand, the question you ask at the beginning is, why did I develop Parkinson's, right? Now, we can give you an answer that we think is an average, and it may never apply to you because you are in a specific category that we happen not to know, because the way we are developing biomarkers is the wrong way. It's the other way around. So, back to our basal model, you can imagine people are in many aspects, many aspects of the pieces that we examined before, and that perhaps there is, right now we know very little where they segregate, where all of us segregate, but if we work the way to understand aging, we begin to tease out sections that relate to individuals with a specific biology, with a focus on the biology about which therapies already exist and are gathering dust in the proverbial pharmacy shelf. Systems biology is what other fields of medicine have done. We do clinical pathology, a 200-year-old concept, because we think that what is in the autopsy of individuals tell us everything we need to know about what happened to them in life. So in Cincinnati, we have begun that process. It's a very difficult and long process, and we're doing it because it's hard and because it is unsexy, but because it has to be done. And we're not classifying, obviously, people are entered with a label. Some have a label of Parkinson's, some have a label of Alzheimer's. But that's not how we're thinking of them. We are beginning with the assumption we don't know what they have biologically speaking. And it's a large study, but it is the kind of moonshot that we need to begin to understand diseases of aging one at a time at a molecular biological level. And maybe when we find these conditions, we probably will be thinking very differently, right? Rather than very large clinical trials, we are going to be in a position when the future clinical trials is not in hundreds, let alone thousands, but in the dozens of patients that happen to have a molecular abnormality that to them is critical and for which we have a treatment. And the question is, and we haven't even touched that yet, are we ready to this phase? Are we ready to a possibility, which is probably the only one that will work, that most treatments to change the disease in people with the disease will work in 1, 2, 3, top 5% of everyone we call Parkinson's. But if we are okay with that, then we are finally embracing what other fields of medicine have done. They begun small because they have large failures. Then they realize, all right, I guess there are more breast cancers than one, right? And then began with a small target and began to spread it. Thank you. So my original thought is, coming back to the first element that I said, is we need you. We are not going to change from within. We just won't. These ideas that I presented to you are not new ideas. When we talk within ourselves, we kind of get it. But we are not going to change until you force the change. Very critical. When you hear the word, we're going to cure Parkinson's, I hope you know that is false. We all want to hear it. We all want to believe it. But with what you now know and with what we all know, there is no Parkinson's, but people with Parkinson's, and we can only cure people with Parkinson's. Any questions? How much time do we have for a discussion? We have at least 10 minutes. Yes. How do you fine-tune it? So there are two levels of treatment. One level is symptomatic, and the other level is disease-modifying. We have no second level. So we are optimizing symptomatic treatments in individuals. We're giving them more levodopa if they need to. We're taking some medications away if they're being harmed by it, for their symptoms. For their symptoms. We still want to tailor to individuals, for sure. But we have to think, even in a more individualized level, if we want to alter their biology, to then move to the next level, which is disease modification. But we're never going to get there unless we accept that there is no Parkinson's, but people with Parkinson's. Yes?
Sometimes I think individuals don't volunteer for clinical studies because there's the caveat that they have to discontinue their medication in order to do so. So if you're basing it on biological markers, does that mean it broadens the base for you to select individuals for clinical studies?
Yes, it does. So if we no longer are thinking that what is going to change things is the symptomatic treatment, then you can stay on whatever treatment you are. But the term broadens is also a little bit problematic, because we're actually trying, if we're going to make progress, we need to narrow, we need to narrow the scope of what is it that we're going to, the who has to be in every trial. And if you looked at trials, they're beginning to, but we're talking about less than 5% of the trials are specifying who the therapy is for. Those trials are for great therapies for Parkinson's disease, not for specific people with Parkinson's disease. Yes?
This systems biology to precision medicine, are you then, when you have these few patients, are you looking at their genetics?
Correct.
Oh, you're looking at their genetics.
And that's the beginning of it. The genetics is the starting point, because people have genes. In fact, the most common Parkinson's gene is the LRRK2. But most often, people don't develop Parkinson's even though they have a LRRK2 mutation. So there are many other factors that can give rise to Parkinson's. And then for those who develop Parkinson's, there are many different subtypes within the same genetic form. So that's why I call it a point of entry. It's very important to know what that genetic entry is, if we can ascertain it. Most often we cannot, and then we go from there. We're not so much trying to stop at the genetics. The genetics as a way to understanding what is it that then affected the biology. Because we have lots of genetic mutations, they may not be acting on us in the way we think they are. And it's a challenge because, of course, genetic abnormalities, we think of them as being determinant. They're not. They're not determinant. It's one of the problems we have with genetic counseling, right? Am I going to pass this on to my children? Well, if it's genetic, you may pass the gene, but not the disease. Creates all sorts of uncertainties, as you can imagine. But there are many genes that could be protective as well. Correct. Correct. But how they are protected, we don't know. We need to actually understand how people in the same disease, in the same genetic family, would have disease and no disease, right? Because there will be people with the same genetic mutation without disease. What is it about those have the gene in the same family and no disease? What might be protecting them? That's a very important line of research. Yes.
We all leave our brains so you can send them somewhere.
So the question,
you have a need for.
yeah, so the question of brain donation is a very important one. And yes, I hope that all of you think about that. It serves tremendously. I wanted to emphasize, though, that there is a limitation as to what we can inform from the brain. It helps many things, but it can help us tremendously. It just happens, though, that the model we have is not systems biology. We have a model called clinical pathology, which means that if you have the pathology, you have the disease, because that fits in with our model. Pathology, we think, equals etiology. Essentially means that which is there in the ashes of the fire is what caused the fire. That assumption, it's a completely different lecture, but there's plenty of data that doesn't show it. In fact, some of the lectures that I've given start by saying, I'm a Martian. I'm going to show you data, and I want to let you conclude from the data. Right? So for instance, just to give you an idea, very simple idea, young people, they underwent a PET scan of their brains. None of them had amyloid in the brain. We think of amyloid as part of what's called now the Alzheimer's continuum. So none of them had it. They were asked in this experiment to not sleep one night. The next day, they had another PET, and guess what happened? It was filled with amyloid. Conclusion of the paper. If you don't sleep, you develop Alzheimer's. But it turns out that these same individuals, actually, there is nothing on that data that would be against the idea that amyloid aggregated in reaction to as a protective mechanism for sleep deprivation, which was the injury the brain was sustaining at the time. Once sleep comes back, guess what happened to amyloid? Goes down. Animal models, mice, we think Parkinson's, because Parkinson's is one thing, we need to figure out where it begins. It begins in the gut, we're saying to ourselves. So there is an experiment where you inoculate the intestines of the mice with E. coli, a bacteria, bad bacteria. What you can measure after is a lot of synuclein aggregations, a lot. So we say here we have a demonstration that Parkinson's begin in the gut. Here's the inconvenient fact, but it's never reported in that way. The inconvenient fact is that if you treat the E. coli, all that stuff goes away. So nothing about the model, again, that tells us that the aggregates of proteins are, in fact, causing. So let me tell you what I'm learning from, may I have like 2 or 3 minutes to explain something that I think is going to bring this point even better. Could I do that? All right. So I have, I was prepared just in case this would come up. I had an agenda here. Turns out that I've learned tremendously. There's a group at Karolinska Institute in Stockholm that they don't have any of the biases we do. They don't think they own the truth like we do. So they look at the proteins in the brain, and they have learned that every protein in the brain that is functioning must be in a soluble state. That's how they are. That's the proteins are very fun. In fact, the proteins are in technically a liquid state. When the proteins get exposed to something that shouldn't be there, and here I'm going to bring my pen, something that shouldn't be in the brain, what happens is that this protein does this stuff. Exposed to it, this is the protein on that. The protein has essentially committed suicide. It is no longer working. This is a thermodynamic mechanism whereby a phase transformation from liquid to solid occurs. A functioning soluble protein becomes insoluble solid protein. It is so stable in this level that cannot do either normal function nor bad function. It cannot even be toxic. But this is the autopsy. Here's where we're looking at the ashes of the brain. We're thinking, oh my gosh, look at this. Look at this. This is bad, right? This must be toxic. Because at the time we're examining the brains, we forgot this stuff, because this stuff, when it was before, has become invisible. We don't see what has disappeared. We don't think what is out of sight. But our entire field of therapeutics for Alzheimer's, Parkinson's, progressive supranuclear palsy, frontotemporal dementia, all of the diseases of brain ages, all of them are based on the idea that there is toxicity of this. It's called a gain of function toxicity. And if we destroy it, we're going to be there. How many trials do you guys know have been published already on this model? The anti-amyloid. You know, we think Alzheimer's is far ahead of Parkinson's because they have so many more things that they are doing, so many more trials. They have already published 35. 35. So we're talking about billions of dollars invested into this. Now, the usual thought about this is that, well, you know, the next trial will do it, right? The next one will do it. What people don't know, and here's where I want you to realize this, people don't know that it's not just an issue of power. If only we had recruited more people, we would have gotten there. 40% of those that received treatment and successfully reduced the amyloid, so this got completely gone, right? Which, by the way, this is what's missing. This is precision medicine. We don't know what this is that has created this to begin with. So everybody that got the amyloid gone, 40% of them were worse compared to placebo, cognitively and by brain volume. But we don't want you guys to hear that, right? Because we know that the story we've been telling is the right story. It's a compelling one, makes a lot of sense, it's unifying, it's logical, it goes from cause and effect. Why would we change it? It's a beautiful story. But it is harming people. So it's not even about the fact that we're investing billions of dollars into nothing. It's about the fact we're harming people. And whenever I'm asked, so how are you doing it? Because of course we're all doing studies, right? We're all doing these studies. I say, my center is not doing them. My center is not doing these studies, and I'm hoping that most centers will begin to stop doing these studies. Because the gain of function toxic model has not supported humans. It's only supporting mice. Mice proteins are really toxic. You inject them with bad proteins aggregates that they never had, they get sick. And if you take it away, they get better. So we really need you guys. We really need you. If somebody tells you on the internet, just the basic level, you're going to find a lot of businesses telling you that the government doesn't want you to know, but I have the cure for Parkinson's. Buy this for $1,500. Don't trust anything like that. You are unique, and no one knows what is it about your brand of Parkinson's and what treatment you need to change that disease in you. And whenever we figure that out, it might not work for others. That's the challenge. That's the challenge. But unless we embrace the reality, we will not make progress. We will continue to waste resources and continue to harm people. We can do a lot better. Thanks, everyone.